So, welcome. This is the Cousin Center for Psychoneuroimmunology seminar series. We have a delight to have Julie Spicer visiting us from Columbia University. She is a recipient of the K99 Award, which is an independent grant, a Young Scholars grant, looking at socioeconomic status and allostatic load, pregnancy outcomes, and maternal behavior. And so she's visiting UCLA, sort of meeting up with old friends and getting to meet new people here. And today, okay, so you can't hear me, okay, fine. So anyway, it's a pleasure. She has, she's currently assistant professor of behavioral medicine and psychiatry at Columbia University. She trained also at Columbia, got her PhD, and before got a BA in psychology at Northwestern University. So it's a pleasure having you visit us today, and we look forward to your conversation and talk.
Thank you for inviting me. It's really a pleasure to be here today. Being someone who works in stress biology and health, it's certainly the case that the faculty from UCLA have contributed significantly to my thinking and development. So I'm really, as I said, really pleased to be here to share some of my work with you, and I also look forward to your thoughts and feedback on it. The title of my talk is Health Neuroscience of Stress, a Lifespan Approach from the Prenatal Period through Adulthood. So what is stress? Stress has been investigated by researchers for decades, if not a century, and there's still much debate over what it is. But for the sake of my talk today, I will refer to three forms. One is social evaluative threat, which is a situation when individuals might find themselves in a place of either being negatively evaluated by others or rejected by them. Perceived stress, which is a self-report of distress or uncontrollable situations in your life. This is something I rely on the Cohen questionnaire, the perceived stress questionnaire, for this. And then also low socioeconomic status, where an individual might not have enough resources to be able to function in daily life, causing them distress, or they might perceive themselves as being lower on the social hierarchy as relative to others. So it can be objective and a perceptive measurement. And the framework that my work typically takes is to think about a stressor, some sort of psychosocial stressor, in association with some outcome like cognition, physiology, or health. And then, given my neuroscience background, I'm interested in asking how this association might be mediated by the brain regions, brain circuitry. So for today's talk, I will cover 4 studies looking at various outcomes. And so for our first study will be the outcome of working memory. The second study will be a cellular level study looking at the outcome of endothelial cell-derived microparticles. I'll explain more a little later what those are. This third study will, in taking a lifespan approach and thinking about women's stress and how that stress during pregnancy might transfer to offspring, I will describe a study that looks at maternal prenatal blood pressure in association with birth outcomes. And then finally, I'll describe a little bit about the K99 ROO project that looks at birth outcomes and maternal caregiving behavior. So the first study involves the outcome of working memory. We're looking at a stressor, a psychosocial stressor, in association with working memory. This study does involve the brain, so I'll describe what brain regions might be driving this response here. To tell you a little bit about our procedure first, we had 2 groups of participants. And they came in for some fMRI scanning. Then our stress group experienced a social evaluative threat, which is classically studied in the lab by the Trier social stress test, where you have, and these were actually our actors from New York who came every session to be our Confederates. But what we really need here are two live Confederates who instruct the participants to come up with a speech that would be self-relevant for that particular person. I'm sure many people here in the audience have experience with this paradigm. So the participants are kind of told by surprise that they need to develop and deliver a speech to two people who are actively evaluating them at the time. And then they were given a surprise backwards subtraction task that is pretty challenging because they're told, at least in our case, to subtract backwards by 13s from the number 1040. And anytime they made a mistake, they were instructed to start over relatively sternly by our Confederates, and also to move faster. So I'll show you our cortisol data in a moment for our group. And then we did have a control group also. And this control group did not meet our Confederates. They were simply asked to sit alone in a room and to do a writing task describing how they would spend the day with a friend visiting New York City. So it was meant to be kind of fun and definitely non-threatening. And by the way, our participants did go through the scanner, some scanning at the beginning, but we pulled them out of the scanner in order to execute this part of the study. Then they went back into the scanner. They had some cognitive testing in between their Trier and in between the working memory. So that was about 30 minutes, and then in between, and then they were given a working memory task, and this task is a very challenging working memory task. It's the NBAC, and they had various levels of difficulty within this challenging working memory task. So first, I will show you our data. These are our cortisol data. In the Trier task, you would expect to see a cortisol effect, and we were looking for that in particular to see whether our Trier, our TSST, whether it was effective. And indeed, let me orient you to our stress group. This is our social evaluative stress or threat group on the dark line, and the control group on the gray line. So at around baseline here, they're about the same. And then as about 20 minutes later, which is where you would expect the bump, we see that our social evaluative threat group increased in their cortisol, whereas the control group did not. And incidentally, the peak is happening just about right before the working memory the challenge occurs. So it seems that cortisol was still elevated while our participants were being administered the working memory task. In terms of the working memory data, just behavioral data alone, this is a relatively simple graph, but it's just to illustrate that we did see a working memory impairment in our social evaluative threat group relative to our control group. This result was derived from a principal components analysis that looked at components, various components of the task, and we found that the component that loaded on good accuracy and short reaction time, which would be a sign of optimal performance, it was the case that our control group was faring better on that. So this was our evidence that we wound up with a working memory impairment effect in our social evaluative threat group. But another thing that we were very, very interested in was what was going on in the brain across all of this. So in order to assess that, we implemented a mediation model. It's similar to the one I showed before as a framework. So we have our stress groups in here, the social evaluative threat groups, or the social evaluative threat group versus control. And we look at that in association with working memory performance. I already mentioned that here we see an impairment of between stress and working memory performance. Then on path A, we'll ask, how does stress activate in the brain? And for this particular analysis, we were doing a working memory versus rest contrast. And when I say rest in this context, it's really the rest that was we had built into the task itself. It wasn't a rest period that we had administered to them, say, at some point before. This is rest that was actually built into the task. And then on path B, you can assess with this analysis, you can assess how does the brain link to working memory performance, controlling for stress. So this is just asking what is the brain doing when it's doing working memory, irrespective of stress. And then finally, so let me just let's see here. Let me just go on to the results. On this path, we saw widespread deactivation of the brain. So stress tended to deactivate the brain in many different regions. And one particular region we noticed was the intraparietal sulcus. That region has been implicated in working memory tasks and in attention tasks in the past. But before we were too excited over that, we had to assess the other paths. So in this path, we saw an activation of the intraparietal sulcus, meaning that the more it activated, the better an individual's performance was, irrespective of what group they were in, stress versus control. To ask what mediates the association between SET and working memory, what we would do is we'll multiply this path A by path B. And so in multiplying this, we get multiplying a negative by a positive, we get a negative. So the mediator appears to be deactivation in the intraparietal sulcus, and bilaterally. So this was interesting because this region had been implicated in the working memory literature before. But we also got another result that was of interest to us. And although I said that we saw widespread deactivation with stress on path A, we did see one region pop out that actually activated. And that region is the ventral medial prefrontal cortex. We were interested in that during a stressor because the ventral medial prefrontal cortex in a variety of studies has been linked to affective self-monitoring, to physiological responses. These are things that might be activated under a social evaluative threat. So to follow up that finding there, we implemented a connectivity analysis. In this analysis, we used the seed regions from the IPS, from the intraparietal sulcus, the ones that I showed you that were mediators. And we put them in as seeds and did a voxel-wise analysis. Under stress, it appeared that there was less connectivity between the IPS and the dorsolateral prefrontal cortex, another region that's very important for optimal working memory performance. So under stress, it appears that a circuit that is generally needed for good working memory performance was not activating together in the optimal way. However, we saw that there was increased connectivity between the IPS and this VMPFC region, suggesting that possibly that region's activation was interfering with the job of the IPS during working memory under stress. So in summary, this is the first study to provide direct neural evidence linking a social challenge to impaired cognition. I say that in part because the studies prior to that, although some of them during a stressor in working memory and neuroimaging, although some of them had the various pieces of data, for instance, behavioral data, neural data, they hadn't linked them directly together in a statistical way. So this is the first study to do that. We see that there's a bilateral intraparietal role for the bilateral intraparietal sulcus, mainly a deactivation of it, leading to impaired performance during stress. We also got connectivity findings with this intraparietal sulcus region, with the ventral medial prefrontal cortex showing increased connectivity with the IPS and the dorsolateral prefrontal cortex showing decreased connectivity. The second study is one that doesn't involve the brain. It's one that looks at social evaluative threat in relation to endothelial cell injury. So this is a cellular level study. And in this study, which was published, we approached it from this point of view. We in collaboration with Dr. Daichi Shimbo and Matt Berg at Columbia, who work on endothelial injury, together we noticed that endothelial injury has been observed under psychosocial stress. This is when the endothelium becomes activated. Although it had been studied has been studied using flow-mediated dilation, by studying endothelial cell-derived microparticles, you can get at the cellular level mechanism, you can get a direct marker of endothelial injury. To illustrate what I mean by endothelial injury, here is a cell, here's an endothelial cell, and when there's a stressor, it might release particles that are indicative of the activation or apoptosis of the cells. And in these particles, there might contain fragments of mitochondrial DNA that are recognized by leukocytes as foreign bodies or even bacterial. And that might spur these leukocytes then to produce cytokines. And this mechanism, or endothelial injury, has been associated with risk for cardiovascular disease. It's important in terms of future health. And as this figure illustrates, it downstream it could be partly an inflammatory process. So the thing about the EMPs, however, is that really up until the point of our study, they had been studied in terms of anger recall, like in terms of psychosocial influence on them. They had been studied in terms of anger recall, but not in terms of social evaluative threat. An added piece to the study is that we had stress group and we had a control group. We had a third group, and I'll tell you a little bit of background on that. This was a group that we intended to be basically an intervention group. We applied a psychological manipulation to this group that could serve as an intervention. And it was based on a self-affirmation of values intervention. This has been shown in previous studies to have efficacy in terms of reducing the racial achievement gaps and increasing good health behaviors such as diet. So it has a rich history, it has a rich literature in other domains. We did this in collaboration with Dr. Valerie Purdy-Vance at Columbia, and we're applying it now to sort of acute stress situation, which was brand new. The affirmation of values, it means that participants were given a list of 7 values. Our intervention group was asked to pick the value that was most important to them and to write about why. The control component to this intervention is that participants are asked to look at the same set of values, pick the value that is least important to them, and to write about why it might be important to somebody else. So for this particular study, we had 2 questions. Does social evaluative threat increase endothelial cell injury as measured by higher circulating and endothelial cell-derived microparticles, or EMPs? And then, does a self-affirmation intervention ameliorate these effects? Our study was built very similarly to the one I just described, although as I mentioned, there is this added group. So we randomized our participants into 3 groups. One who got the social evaluative threat plus the intervention, one who just got the threat alone, and then a control group who got neither of those things. So this group got the values affirmation, while these 2 groups got the control task. Then these 2 groups got the stressor, and this group got the friend task that I described earlier. Across time, we were drawing blood samples in order to get blood plasma for the EMP analysis. And we got the blood samples at 3 different points. So this was one prior to the stressor and then at 2 different points afterwards. So I'll show you the data now. We had 3 EMPs. As I mentioned, they can be indicative of activation of the endothelium, apoptosis, or both. So I'll describe in detail the results from one of our EMPs. This is our intervention group, and first let's look at just the stress group alone on this solid line. So over time, from baseline up into I guess what you would refer to as the recovery period, we see an increase. And then with our control group, which has these elongated dashed lines, we don't see at all an effect. And then the question becomes, what happens to the intervention group? Do they look more like the stress group? Do they look more like the control group? So with the short dots here, we see that our intervention group looks more like controls. We had 3 EMPs in total, and the other 2 shown here, they basically they have the same pattern as the first one. Because this was a relatively novel study, we also reported we had other measures that had been built into it, and we reported on those, the more classic measures for social evaluative threat. So we had salivary cortisol, and we had self-report of psychological stress. And although they don't match the EMP data like directly, they did fall into a pattern that makes sense to us. So in this way, we feel that they corroborate the novel EMP data we have. In the way they make sense to me, to make sense to us, is that with our stress groups, we get an increase in salivary cortisol. With our control group, we do not. And same goes for the psychological, self-report of psychological stress. We see that they both increase, whereas the control group does not. And I'm happy to explain this later if there's interest. That makes sense to us too. So in summary, what we found was that social evaluative threat increases endothelial cell injury, that self-affirmation intervention attenuates this response. And one thing that was interesting to us was that it's a situation where there are basically like 2 psychological influences on stress biology. There's the social evaluative threat, and then there's also the values affirmation intervention. So one psychological process was able to essentially block out the other one, and then with the effects wound up being on biology. And then this could be important because EMPs have been linked to endothelial function has been linked to cardiovascular disease risk. Now turning to some work that I've been doing in the last couple years. It takes a lifespan approach, and it's work that is built into the K99 ROO project that I'll be carrying forward for the next 3 years or more. And this lifespan approach looks at women's stress, maternal stress in the perinatal period. It's interesting in that the work, well, for one thing, maternal pregnancy stress has been shown to influence pregnancy outcomes, including gestational age at birth and birth weight, the quality of maternal caregiving, which is very important for offspring development, and it can affect that offspring's health and achievement across the lifespan. And also, it makes, because of these effects, it makes understanding targets for prevention and intervention in this period of vulnerability quite important. Another interesting point, at least to me, about studying this period of time is that it takes 2 participants together, thinking about the dyad, the mother and the infant, and not only that, but thinking about how their interaction basically shapes the next generation. So my first study that I'll describe that falls into this type of work is one that looks at ambulatory blood pressure trajectories and stress in normotensive pregnant adolescents and its associations with birth outcomes. This is a work that's in progress. So back to the model from the beginning, this would be taking perceived stress and looking at it in relation to maternal prenatal blood pressure, and asking what if there's an association with birth outcomes. It makes use of a finding that does show up in the literature, whereby across time in gestation, in pregnancy and gestation, there is a U-shaped curve. A U-shaped curve has been identified in blood pressure. And not only that, but a flatter U-shape has been associated with adverse pregnancy outcomes, including pregnancy and intrauterine growth restriction. In terms of stress and birth outcomes, there have been a couple studies out there, put out there. One is, with its findings, it's been shown that pregnant women who demonstrated more stress reactivity delivered lower birth weight infants. In a different study, a composite measure of stress, this includes accounting for socioeconomic status, perceived stress scale, and anxiety. This composite measure of stress interacted with diastolic blood pressure, such that the higher the stress measure and the higher the BP, the lower the birth weight. And in another study, lifetime racism interacted with diastolic blood pressure, leading to lower birth weight. So there is some evidence to suggest that blood pressure does interact with stress to produce poor birth outcomes. So for this study, we asked two questions. Will higher perceived stress be associated with a flatter U-shape in normotensive participants? And will a flatter U-shape be associated with poor birth outcomes? In our case, we're going to look, in terms of the data I'll show you today, we'll look at gestational age at birth. The study procedure, just briefly, this schematic shows that we followed these participants over pregnancy and saw them 3 times. One in early pregnancy, and one in middle, and then one in late pregnancy. Over these visits, we collected multiple mood and biomarker data. And then finally, at the delivery, at the birth, we were able to get from the medical chart the gestational age and the birth weight of the infant. In terms of our statistical approach, we were interested in this U-shaped trajectory. And in order to assess it, we looked at 2 important statistical parameters. So one is looking at just at gestational age. It's a linear term. It's going to get at the dip that goes from the beginning of pregnancy into middle pregnancy. Then the second term is a quadratic term. So it's gestational age squared. And it's getting from mid-pregnancy up until the end, it's getting at that acceleration. So we have 2 parameters that make up the U. In terms of perceived stress, this is something that we had expected to find and did find in preliminary analyses, although I have to say that it did fall away in recent analyses. So we're less sure that stress did interact on the trajectory, but in terms of finding a flatter, or in terms of the low stress, we did see something originally with the U-shape, and then a flatter line for the high stress. Although unfortunately this finding does not seem to be holding up right now. One thing though that is of big interest to us in the project does involve the birth outcome results. And in order to assess this birth outcome result, we turned to this paper from a few years ago, published in JAMA by Kathy Spong, who's at NICHD, who argued that although gestational age at birth in many studies and even in the clinic has long been thought of as a binary thing, where there's preterm that comes before 37 weeks, and then term that comes after 37 weeks. She argues in this piece that and it's along the lines of every week matters in gestation, and she argues that there's not just preterm and term, but in fact 4 different ways of assessing term: early term, full term, late term, and post-term. And these all map out post 37 weeks and beyond. And even something that's 34 to 36 weeks would be considered late preterm. So in our analysis, I would like to note that we use gestational age at birth as a continuous measure. We didn't break it down into categories, but in order to visualize the data, we utilize this paper. So the first thing that we saw, and I should also say that these findings control for stress. So these are holding up irrespective of the stress measure. So the first line I'd like to display is this one. It's at 39 weeks gestational age at birth, which actually it falls into the full term category, and it is also the mean for our sample. And then the question is, does the line does it become flat once gestational age at birth decreases, which is what we would have expected from previous literature? But in fact, what we see is that it becomes more pronounced, and then in late term, it actually inverts. So in sum, this is the first study to examine pregnancy-associated U-shaped blood pressure curve as a function of perceived stress. And we found that increased perceived stress over pregnancy was not associated with a flatter U-shape. However, with respect to gestational age at birth, although there was no interaction with perceived stress, but it looks like an optimal U-shape may index late full term. A stronger U-shape may link to early birth, and a lack of the U-shape might link to later birth. I show the image of Goldilocks here because with the idea that it's about not too much and not too little, but finding that U-shape that is just right for optimal birth outcomes. So in the last few minutes, I was planning to leave some time for discussion and questions. And in the last few minutes, I would like to walk through a little bit of what I'm doing on the K99 ROO project, which is entitled Socioeconomic Status and Allostatic Load, Pregnancy Outcomes and Maternal Neurobehavior. This project follows women across a pregnancy period and into the postpartum. And excitingly for me, it's one that looks at stress biology as a function of allostatic load in collaboration with Dr. Tressa Seaman, who is here. This is one of her early papers on allostatic load. And just briefly, allostatic load is considered a multiple physiological index of dysregulation. So it covers some of the systems I've already mentioned today, like cortisol, the HPA axis, and blood pressure. It also it can cover inflammation, metabolic ones, cholesterol, various ones, to get an assessment on how the allostasis in one's life has contributed to a load that in and of itself can be linked to health in different points in life. So, but the thing is that it has been relatively understudied during pregnancy. So what I had aimed to do was first to assess socioeconomic status in pregnancy in association with allostatic load, and then to ask if these things affect birth outcomes, with the idea that we think lower socioeconomic status will be linked to higher allostatic load and then play out in terms of poorer birth outcomes. For this, I was excited to partner with a population-level data set of 10,000 women followed prospectively through pregnancy and well-characterized for biomarkers and psychosocial markers, with also birth outcome data. Certainly, I can't assay allostatic load on all 10,000, but we were able to take a subset that that were enriched for early birth. So we'll have like a nice range of gestational age at birth and birth outcomes. And we'll get to do a 10-plex cytokine panel with them, with the serum that was collected. And in total, it'll be about 900 women who will go into this analysis. We're in the middle of the assays at the moment. The second part of the grant, these two things I've been already working on, and the second part of the grant is a laboratory study that aimed to assess socioeconomic status in association with low allostatic load, and with the outcome being maternal caregiving. This was I can achieve this type of thing because there are excellent data sets already in the lab of my primary mentor, Catherine Monk, on the grant, and with collaboration with Dr. Amy Hain at Williams. So in the lab, women were followed and biomarkers were collected during their pregnancies, and then they come back at the 4-month mark with their infant, at the 4-month postpartum mark with their infant, and they do some tasks that allow us to do behavioral assessments. So we're interested, in order to capture maternal caregiving, we are utilizing a 10-minute free play that occurs between the mother and the infant that is videotaped, and then working with the Ainsworth criteria on which Dr. Amy Hain is an expert. We're trying to extract out from those tapes measures of maternal sensitivity, maternal intrusiveness, for example. And the idea is, the overall idea is to ask if stressors during pregnancy of this nature lead to poor birth outcomes, or do stressors in pregnancy lead to poor maternal caregiving. Finally, this is something I haven't gotten a chance to start yet because this will be for the next 3 years, but this part will assess allostatic load during pregnancy, and then in the early postpartum, still around the 4-month mark, I will get measures of maternal caregiving, just as I had described here. Also, excitingly, we get to do some brain scanning, where we will ask the mothers to go into the scanner, and at the moment, the plan is to administer a task that is comprised of infant stimuli, specifically infant cry, and this is with a precedent in the literature, and we expect that we will get some amygdala and anterior cingulate cortex differences as a function of their pregnancy stress. We don't have a good task at the moment to get at maternal interaction with her infant in the scanner, but what we can do is we can take the brain activation from the infant stimuli task in the scanner and do some correlation with the behavioral measures that we'll get within like a day or two of the brain activation. So with that, I'm happy to take any questions, follow-up questions on this, but with that I will acknowledge the people who have contributed to the work and contributed to my career development. This includes Catherine Monk, who is my primary mentor now at Columbia, Tor Wager, and the late Ed Smith, who were my PhD mentors at Columbia. A whole number of people who've been mentors and former mentors and current collaborators, and also these funding resources. Thank you.
Questions? Liz?
looking at that last slide. I think you said you had multiple biomarker measurements, and maybe you don't have this data yet, but how much within a given woman does the allostatic load change over pregnancy? And in a model like that, where you're putting allostatic load as if it were a single measurement, how are you dealing with that? Or how do you plan to deal with that?
Yeah, that's a really great question, and it's certainly been on our list. You know, to think about it's we do have multiple visits in pregnancy, so we have 3 visits. Let's see, we have 3 visits collected in each of these studies, and I plan to do multiple visits in this study. I have had some conversations with Teresa about it, and really it's first of all, we don't know much about allostatic load in pregnancy, so physiology is already moving around in pregnancy, and this work will hopefully contribute to the knowledge about how allostatic load behaves in pregnancy and whether it's an important marker for health overall. And then we haven't gotten to the point where we've had to make a decision about it, but we've come up with different ideas. Like we could take an average of the allostatic load across pregnancy. We could take the highest one, but that we find, but we haven't, gotten into the data yet to know what is, really where the signal is.
And then the other question I have is, so the paper that says a week matters, a week's different matters, what's the starting point, and how confident are you of that starting point for determining the number of weeks, the gestational age?
Meaning like from category to category, or just? How
is that determined? How do you know that someone is 34 weeks or 35 weeks or 36 weeks?
Yeah, right. So it's determined, we determine it taking the marker of the last menstrual period, and then also we can, prior to the pregnancy, we also I believe, use that in combination with ultrasound as the pregnancy progresses, using the two together.
Do you know how much variability there is? I mean, if 1 week makes a difference, the recall of the last menstrual period, and there's biological variability in the ultrasound. So I'm just raising that. If you're trying to make these fine distinctions at the end of pregnancy, and your birth outcome is completely dependent on the value of that gestational age, what's contributing to the uncertainty of that value?
Right, that's a very good point.
Paul?
Yeah, I had a comment. I've been looking at this maternal caregiving work that you're embarking upon. There's somebody who it didn't occur to me that you might want to meet here, whose name is Ariana Anderson. I don't know if you're familiar with the work that she's done on developing machine learning algorithms to look at baby cries and use those and develop expert classifiers to know when babies are distressed. So anyhow, both for evaluating maternal behavior and for certain development of certain tasks. She also happens to be an expert in dynamic causal modeling and fMRI, and teaches the summer course here. So she's into both the imaging side and also baby cries as a part-time job. So
that would be great. That would be really great.
As a collaborator.
Thank you, Paul. I have a question on the allostatic load. So what aspects of the allostatic load do you think are contributing to changes in maternal caregiving?
So this is really novel work, so I really don't have the answers yet, but part of the thinking in looking at maternal caregiving dated for me back to the animal literature that has looked at stressors during pregnancy and caregiving, and then there is some work looking at socioeconomic status in association with maternal caregiving, but I'm not sure that it followed, like to look at it across the pregnancy phase. That's relatively new as well. And I would appeal to the animal work in terms of answering your question. I would think that there could be a cortisol influence. One thing that we did propose to do was to not only look at allostatic load alone, but to look at these biomarkers, or not to look at allostatic load solely in these models, but also to look at the biomarkers that contribute to allostatic load alone, to see if there's any particular one that's like driving the responses. With the sample size from the first study, we should have.
So you had mentioned that you're also going to be looking at inflammation, inflammatory markers.
Yes.
What evidence is there that there's a link between inflammatory markers and maternal caregiving? I certainly know it's related to birth outcomes. How about maternal caregiving?
There's very little to my knowledge at the moment.
I was wondering, in the graph where you're talking about like the PSST, and there was the endothelial derived cell markers, and there's also a cortisol level, self-report stress. Self-report stress, the control group wasn't stressed with PSST, and they were stressed later.
Yeah.
And I was wondering why that was.
So I don't have direct evidence for it, but we had an idea about that. We think that it's because the control group on the whole they had it relatively easy up until that point. They were going through some scans, they were at rest, they were doing the friend task that I described. And then at that point, they had all the groups had cognitive testing administered to them. I would guess that the control group found that cognitive testing, our working memory tasks and otherwise, they found an increase in their stress at that point because they actually kind of they were challenged in a way that they hadn't been challenged before. Whereas our stress groups, they were already pretty stressed out, so they didn't find the cognitive task to be like, it didn't make as much a difference to them. That's my thought.
For study 3, I noticed in the titles that the moms that you studied were actually adolescents themselves. Is that correct?
Yes.
So does that influence how you interpret the results? Because if all of the pregnant people were adolescents themselves, is it generalizable? Or has that work been done in either young or not middle age, but like women in their 20s and 30s during their pregnancies? And why you selected adolescents as that?
Yeah. So thank you for the question. I didn't get into all the details. So one thing I didn't mention was why did we select adolescents for this project? And one reason is that although we don't love the terminology, you can think of them as a group that, if we're interested in studying stress and the effects on blood pressure and birth outcomes, you can think of them as a group who might be enriched for stress, because pregnant adolescents tend to have certain backgrounds that are, made with a little bit more adversity than non-pregnant teenagers and or women who are older. So that's one reason why we focused on adolescents. And then in terms of looking, so I don't think that certainly a follow-up study would have to be done to see whether these patterns are playing out in other populations. The UShape I showed, that other work has been done in women who are, have a range in age. But what I find interesting in thinking about adolescence and pregnancy in adolescence is, in addition to all of that, the fact that, for one thing, they're young on the scale, on the scale of becoming pregnant, and they should be like relatively young or relatively healthy individuals overall because of their youth. And let's see, but then there's also this interesting aspect that they're still developing too. So we have to sort of acknowledge all of those things when we are interpreting our data, and certainly they don't generalize always.
Well, thank you very much, Julie, for a very
Sure.
interesting lecture.
Thank you. Thank you.